Hi, everybody. Thank you for attending today's webinar, Count Cells Without Slides, CellDrop Automated Cell Counter. I'm Rachel Kelly, a marketing specialist at DeNovix. I'm here with David Ash in the DeNovix headquarters in our laboratory, and I'm going to be the moderator for today's webinar, as well as our question and answer session at the end. Today's webinar session will be recorded, and we will email a link to the recording after the session. I'm now going to hand it off to Yoon Jin from Phyla Korea, who's going to introduce today's topic as well as our speaker.
Thank you, Rachel. 오늘 웨비나에 참석해 주신 여러분께 감사 인사드립니다. 저는 필코리아테크놀로지 조은진 부장입니다. 저희가 오늘 준비한 웨비나는 디너빅사의 오토메이티드 셀 카운터인 셀드랍입니다. 셀드랍은 소모성의 플라스틱 슬라이드를 사용하지 않고 세포수를 측정할 수 있는 오토메이티드 셀 카운터입니다. Today's speaker is David Ash. 오늘의 웨비나 연사는 데이비드 애쉬입니다. 데이비드는 생명과학 분야에서 20년 이상 몸담아왔으며 특히 유전체학, 분자생물학 및 세포학에 대해서 전문 지식을 가지고 있습니다. 데이비드는 2012년부터 디너빅사의 어플리케이션 디렉터로 특히 셀드랍 개발에 핵심적인 역할을 하였습니다. 그럼 지금 델라웨어주 웰밍턴에 위치한 디너빅스 연구소에 있는 데이비드의 이야기를 들어보도록 하겠습니다. Hi David, please your presentation and live demo first.
Hello, and thank you for attending our webinar today, Counting Cells Without Slides on the DeNovix CellDrop Automated Cell Counter. So for our agenda today, I will touch upon some key features of the CellDrop. I will go into great detail on the DirectPipette Technology and why it affords us to count cells without slides. I will then walk through a brief live demo of how to count slides on the CellDrop. And finally, I will take a look at some applications we find very interesting where the CellDrop is being used today. At the end of our webinar, we will have a question and answer for about 10 minutes. So please load us up with questions. And if we don't get your question answered on this webinar, we will be sure to follow up via email so everybody's questions get answered. So a little bit about DeNovix. We were founded in 2012, and we focus on innovative, unique laboratory equipment for the life science sector. Since 2013, 2014, we have installed thousands of instruments worldwide with our UV-Vis microvolume photometers, fluorimeters, and now cell counters. Our products have shown up in thousands of citations and are trusted by scientists worldwide. So before CellDrop was launched, there was two major ways that researchers would count cells. It would be either the manual cell counting hemocytometer, which has been around for probably 60 years, probably 70 years. And it would be loading a sample on a glass hemocytometer slide, loading that on a microscope, manually counting your cells, and manually calculating your cell densities and viability. About 20 years ago, automated cell counters showed up in life science laboratories. The great thing about them was the automatic image capture, as well as the automatic calculations for cell density and viability. Those cell counters still needed a one-time use plastic slide for usage. In 2019, we launched the CellDrop. So some of the key features of the CellDrop is the primary benefit would be the patented DirectPipette technology. I'll go into more detail about the DirectPipette technology in the next slide. We also have a cell counter that has dual fluorescence capability. We can do bright field imaging as well as dual fluorescence measurements, both green fluorescence and red fluorescence. This instrument does not need a computer. It is controlled by a 7-inch touchscreen, a nice, brilliant, bright screen that allows you to see cells in high definition. You can see on the left, there is a cell count of hepatocytes. We are very happy to announce that we have a hepatocyte counting application that is using a machine learning model. We trained this model on thousands of images on multiple species of hepatocytes, and I'll talk about that a little bit later in our application section. This instrument is also highly connected. You can connect via Ethernet or Wi-Fi, which will allow you to send data by either email to network folders, to network printers, or you can save to a USB drive. So a little bit more about our DirectPipette surfaces. As you can see on the left-hand side, there's a picture of our instrument with the sampling arm in the open position. And the arrows will point to the upper surface and the lower surface. These two surfaces are sapphire, optical grade sapphire, and are very robust and are fixed into the instrument permanently. So there's no need for removing these surfaces or replacing them. They will be there for the life of the instrument. If you put the arm down in the closed position, it will rest on a micro adjuster screw, which holds the upper arm at a specified micron chamber height. For hemocytometers, the typical chamber height is about 100 microns. That is also the same chamber height for plastic slides. What we have here is an arm that carries the upper sampling surface and is allowed to lower the chamber height to a 50 micron chamber height or raise the chamber height to a 400 micron chamber height. And we also have a 100 micron chamber height for standard cell counting applications. These surfaces can be wiped clean after the measurement. So as you see here, the arm is in the down position. The pipette tip delivers the pink sample. A measurement is taken, and then the arm can be picked up into the open position and wiped clean, both the upper and lower surface. So what benefits do we have with the DirectPipette technology? Well, one, it's environmentally friendly. We are reducing the amount of plastic that needs to be purchased, used in different workflows, and disposed of. Also, it provides us the variable chamber height mentioned in the prior slide. The table below will show the different micron chamber heights, the volume required to make a cell count on those chamber heights, and below that is the cell density per chamber height, either for the 4x version of our cell drop, 4x magnification that is, or the 10x magnification cell drop. This QR code will send you to our website, which will allow you to take a look at the cost savings that you might see if you were to use CellDrop in your laboratory. The top left-hand corner will be a slider, which will allow you to input the counts or the cell counts per week that you perform. And you get to select the currency and then also the weight of plastic that will be displayed below. As you can see, once you've selected a position on the slider, below you will see the per year cost savings, per 5-year cost savings, as well as the number of slides saved. So I invite you to please take a look at this and give it a go. So our DirectPipette technology does not require a plastic slide to be used. But in the event that a user would like to use a plastic slide for their application, one can be used on the cell drop. The user will just opt for a slide-based count in their protocol. Some applications where this would make sense would be for samples that are higher BSL rating, that the user does not want to touch the reusable surfaces. Another application that we've seen in the last year or two is for nuclei isolations. You can count nuclei isolations on the DirectPipette surfaces, but if you would like to use a slide for your nuclei counts for your cell density, you can actually transfer that slide used for the counts on the cell drop to a higher magnification microscope and inspect the nuclear membrane or chromatin integrity using a higher magnification objective. So now we'll step into our live demo. What I have here is acridine orange propylium iodide, and here is our home screen. We have several different applications which I'll get into in later slides, but for now, I'm going to use the primary cell AOPI. Touching the application launches the live preview. I'm going to take an equal volume of AOPI stain and add it to our sample. After pipetting up and down 3 times, the incubation time is very fast. It's about 20 seconds, maybe 30 seconds. So by the time I get my pipette, I set it to 10 microliters because I am using the standard 100 micron chamber height. I can load it from left or right. Here, I'll load it from the left with the arm in a down position. Once the cell sample will deposit on the lower surface in about 10 seconds. By the time I dispose of my tip and set my sample down, the sample should have deposited on the lower surface. Here you see the bright field image. Since this is a fluorescent assay for both total cell count and viability, I'm going to be focusing in the green channel. But here you can see the sample settling. And this is actually a blood sample that has been diluted down, where you can see there is a lot of RBCs in the field of view. And what we're staining for is the nucleated white blood cells. A couple of different tools that we have on the live preview is sample information. Should you want to optionally input sample information, you can do that here. This is also a protocol-driven device, which means you're going to select different counting parameters to make the cell count. What we have here by default is a default protocol established in the factory. This default protocol will get you about 95+% to the cell count. Should you need to modify these parameters, you can do that by hitting the plus sign here. This is where you can actually modify the counting parameters and make your own custom protocols. But since I know the default protocol will work for this sample, I'm going to use the default protocol now. So for a fluorescent cell count, I will change the channel to the green channel. And as you can see, the white blood cells are showing up in green here. I will show you the result in a minute. But since I am using one surface and not a plastic slide for all my measurements, the DirectPipette technology allows me to focus once and basically use that focus for the duration of my measurements, unless there is a drastic difference in cell diameter between sample to sample. We also have autofocus. Again, since we're only using one surface on this instrument, if you use autofocus one time, you're good for the duration of your cell measurements. You don't need to use autofocus per measurement. As well as exposure. You can set your exposure, hit OK, and you're ready for your cell count. Once I hit count, in about 8 to 10 seconds, you're going to see the result window show up. And here, what we see is the numerical data on the left-hand side, prioritized from live cells per mil and dead cells per mil, total cells per mil, and percent viability. On the right, you have the result image, which you can actually see, zoom in on, and actually see that the acridine orange propylium iodide mixture fluorescent stain is actually picking up the white blood cells and ignoring all of the RBCs in the background. A couple of other interesting tools that you can use on this top bar right here, toolbar, is the overlay, where you can look at individual raw images or the final result image all the way to the right. You can also export the information from the CellDrop. By clicking the export icon, you can export raw data from the CSV. You can export the result image along with the raw images that go along with the result image. You can also create PDF reports if that's of interest. One last tool that I'd like to show users is the dilution guidance, this icon on the right. What this does is, this is a convenience for the end user. In the current concentration cells per mil, you'll see it's the live cells per mil populating that area. What I can do from here is put in numbers of target cell density and target volume, and hit OK. And what you'll have just above the gating mechanism is your dilution guidance. This dilution guidance will follow all of the result data and be saved on board the instrument once the arm is lifted and the sample is wiped off. So if I want to remove the sample, I'm going to pick the arm up. I'm going to grab a laboratory wipe. Just a general laboratory wipe is fine. I will square it up and wipe in one direction, the lower surface, the upper surface, and close the arm, and the instrument is ready for the next sample. And that's the end of our live demo. So within the CellDrop family, there are two measurement capabilities that you can select from. You can select bright field only, which would allow you to do trypan blue, erythrocin B viability measurements, or total cell counts with you on cell culture samples or samples that have very low to no cellular debris. But if by chance you do have samples that have cellular debris, or if you're using primary samples, or if you'd like to see GFP transfection efficiency, then the capability on the right or left, pardon me, the bright field plus fluorescence is the way to go. Within our measurement capabilities are two CellDrop models. We have the 4x magnification standard magnification unit, either the FLI for fluorescence plus bright field, or the BF unit for bright field only. If by chance you're doing yeast samples, or you're doing very small stem cells, you can opt for the 10x magnification models, the FLXI for fluorescence plus bright field, or the BFX if you're, again, doing cell culture or yeast total counts. So what I'll do is go over the different applications on the home screen. The top level is going to be our applications, which are general applications. They're going to be bright field for total counts, trypan blue or erythrocin B for cell culture maintenance viability or samples with low debris viability. Erythrocin B works just like trypan blue. It stains dead cells and is excluded from live cells. And the erythrocin B is actually a less toxic stain than trypan blue. So it's a good alternative if you're looking for something like that. AOPI is a mixture of acridine orange, propylium iodide. And what that does is stain nucleated cells, live cells green, and dead cells red. It's exactly like the application you just saw in the live demo, except for it's a more general protocol used. Acridine orange used by itself is a good stain for very, very low density samples. And propydium iodide used by itself is a fluorescent alternative to the trypan blue or erythrocin B viability staining systems. So our second level is a more advanced application set. What we have is, we have a few applications here that are double asterisk or double starred. Those I'll go into more detail with in subsequent slides. But going from left to right, we have a GFP transfection efficiency application. We have a yeast application that, depending on the magnification, you can use either FDA PI for the staining of yeast, or if you use the 10x magnification fluorescent unit, the FLXI, you can do both yeast total counts using bright field or yeast viability AOPI staining. We have the hepatocytes app that I mentioned earlier for hepatocyte counting and viability. This uses a machine learning model, and this has been already established. You don't have to train it yourself. We have trained the model on many different species with thousands of different images to get the best counting model for hepatocytes we can provide. Primary cell AOPI is the actual application you saw in the demo, and that will do acridine orange propydium iodide staining of white blood cells or primary cells, which will help exclude any kind of debris that would provide inaccurate measurements. Nuclei AOPI uses the same AOPI formulation as the other applications I mentioned. And what this will do is, if you isolate nuclei for, say, single cell sequencing or single cell RNA-seq, you can use this staining procedure to stain nuclei with propydium iodide. And what the acridine orange gives you is the ability to measure, detect any kind of residual live cells in your isolation. And finally, the fixed samples PI. This is a new application as well. And this really is geared toward RNA-seq or single cell, single nuclei RNA-seq for fixed sample workflows. Our last level of applications, as you see, is the utility applications. There is a custom method counting application, which is a generic application, which will allow you to measure within the capability of the instruments, whatever you would like to measure. The user accounts for establishing protocols for specific users and user accounts that they can log in and actually have those protocols prioritized for their account. We have a data app that will allow you to access the historical data that's saved on board the instrument, either from 1 week ago or 1 month ago. We hold many, many samples on board. We have a settings app that will allow you to take a look at the languages within the available languages within the instrument. It will also give you instrument-specific information. We have a 21 CFR app, which allows GXP compliance-ready systems, if your laboratory would need that. And lastly, we have an updater application. This is just for if we send out periodic system software updates, you can update it from this application. So let's get into some of the more advanced applications. So this is the one I mentioned before. This is our first machine learning application. It's for the challenging cell type of hepatocytes. This community of users that need hepatocyte counting has low confidence in different counters, automated counters. So they opt to do a lot of their counting on manual hemocytometers. What we at DeNovix has done is to create a machine learning model from, I think, about 5 different species, as well as using thousands of different images of all different densities. And if you stain the hepatocyte samples with acridine orange propydium iodide, you can get a viability of your hepatocytes. You can get a live hepatocytes per mil, as you see on the right-hand side. This is a typical result. And you will get a viability of your hepatocytes, as well as detection of other non-parenchymal or cellular debris. As you can see in the right image here, you are seeing little white or gray areas, that's cell debris. And the large green and large red cells are going to be your hepatocytes. I mentioned yeast AOPI for our higher magnification FLXI cell drop. This is going to be good for the brewing industry, or researchers are looking for yeast-specific cell counts. We can use the AOPI staining system with the 10x magnification because it collects more light, and we want to use the AOPI for yeast viability and total counts because it is a faster staining system. And to the right, again, you're going to see the same general format of all our result windows, which is left-hand side prioritizes the numerical data which the users are most interested in, as well as the result image. This one, as you can see, has been zoomed in quite close. So pinch and zoom is an option for the result images as well for a closer inspection. Nuclei AOPI, which I had talked a bit more about in the preceding slide. This is for fresh isolations of nuclei, which may have residual live cells in them. When we use acridine orange and propydium iodide to stain a fresh isolation of nuclei, we can get a nuclei density, and so nuclei per mil. We can also get an intact cell density, which will provide the information to calculate an extraction efficiency. An extraction efficiency is how many live cells are still floating around in your sample. I think 10x Genomics has a recommendation for less than 5%. So you can use this information to gauge the quality of your sample, of your extracted nuclei. And lastly, the fixed nuclei application works almost exactly like the fixed cell application. It just depends on what you're staining for in your sample. So for example, our fixed nuclei PI is going to be stained with propydium iodide. These are going to be fixed cells or fixed nuclei, so they're going to be all dead, and they're going to allow propydium iodide to stain the cross-linked RNA within these fixed cells. There is some special images and information that goes along with this cell type or this sample type. Mainly, we're going to distinguish unclustered nuclei per mil versus the clustered nuclei per mil, and provide a percent unclustered value for the sample. And what that does for the user is tells them how many nuclei do I have free of clusters, so monodispersed nuclei. And if I remove those clustered nuclei, what is the concentration of unclustered nuclei per mil that will probably end up in my library preparation? These types of information are good for parse biosciences workflows, 10x Genomics Flex kit workflows, and scale biosciences workflows as well if they're needing fixed nuclei densities as well as clustered information. One very important image that we've included in these apps, both the fixed nuclei PI and the fixed cell PI, is the color coding of the clustering image, which will allow you to see which nuclei we've counted as singlets and which nuclei we've counted within clusters. And with that, that is the slides that I have. We're open for questions if you have any. I know we have a couple of questions already in the queue. So please, if we don't answer your question now, we will definitely get back to you within email. Thank you.
All right. Thanks so much, Dave. Yeah, so we have a couple of questions for you. And like Dave said, if anybody else has questions as we go along, feel free to put them in the Q&A panel. So the first question we have for you is, how do I know when to use fluorescence versus a colorimetric viability stain?
So that will be sample dependent. If your cell samples are coming from cell culture and they typically have low to zero debris floating around, trypan blue or even erythrocin B is a fine stain for doing total counts and simple viability measurements. If your sample is coming from a primary cell source, or has a lot of debris associated with it, kind of like the PBMC sample that we measured where the white blood cells were surrounded by red blood cells, and we really want the accuracy with fluorescence, that's going to be the best choice.
All right. Thank you so much. So the next question we have is, what parts of the cell drop might require periodic maintenance?
The sample surfaces are very robust, so they're not going to be negatively impacted by using, say, ethanol. But really, if someone were to leave a sample on the sample surfaces, you can use ethanol to remove that sample. You can clean them periodically if you'd like. There is really not much maintenance that has to go along with the cell drop. We've actually developed the instrument to be this way. Upon boot up, we definitely want you to follow the prompts about cleaning the surfaces and making sure the surfaces are free of anything so it can do its auto calibration, and that will give you the best accuracy.
Great. Thanks so much. It looks like maybe the last question, unless we get any more questions in, is what are the decontamination methods that you can use on the cell drop?
For decontamination, you can use either 70% ethanol, 70% isopropanol, or 10% bleach. The detail that you want to follow, though, is when you're decontaminating, say, the entire instrument or the upper platform or the surfaces, you want to spray the ethanol or isopropanol or bleach on a paper towel or laboratory wipe and wipe the instrument down. We don't want to douse the instrument with liquid or dump liquid on top of it because it's not liquid proof. So you may get some liquid down in the crevices, which is not going to be easy to get out. But wiping it down with a lab wipe and ethanol, bleach, or isopropanol is just fine.
All right, great. Thank you very much. And that brings us to the end of this webinar. So I want to thank Dave for his presentation and for answering those questions, as well as the Pilot Korea team. Like we mentioned at the beginning, this whole webinar has been recorded, and we will email everyone the link to the recording within a few days. And now I'm going to hand it back to Yoon-jin at Pilot Korea to close out the webinar and also discuss this post-webinar survey they have prepared for you.
네, 오늘 웨비나에 참석해주신 모든 분들께 다시 한번 감사 인사드립니다. 필코리아 테크놀로지는 데노빅스사의 셀 카운터 브라이트 모델과 애플 아이 두 가지 데모 장비를 모두 구비하고 있으니 많은 관심 부탁드립니다. 화면에 보이는 QR코드를 통해서 설문지를 작성해주신 분들께는 저희가 감사 인사로 커피 쿠폰도 제공해드리고 있으니 많은 참여 부탁드립니다. 궁금한 사항이나 요청 사항 있으시면 웨비나가 끝난 이후에도 언제든지 말씀해 주십시오. 데노빅사 셀드랍 오토메이티드 셀 카운터 웨비나는 이것으로 마치겠습니다. 감사합니다. Thank you, David, and thank you, Rachel.
All right, thank you. And thank you everybody for attending. Have a great rest of your day.